Hello and welcome to another Dementias Platform UK podcast. I'm Josie and I'm the Communications Officer for DPUK. I'm with Professor James Rowe, who's here to talk about the Experimental Medicine Incubator. So could you start off by explaining what the EMI is?
Yes, fantastic. Yes, hello, Josie. So the Experimental Medicine Incubator, it's a partnership between academic institutions, universities with expertise and interest in dementia, and industry partners, including pharma companies and other industry partners, who jointly want to come together to meet those challenges and overcome the obstacles to fast track new treatments for people with dementia or people who are at risk of later developing dementia. So the incubator, the idea there is it's a way to bring these groups together, and by close working and exchange of ideas and knowledge, often materials and really cutting edge information and treatment options to accelerate this. And as an incubator, it's to help grow and mature that solution. So this is not to deliver clinical trials, but as an incubator, it would get compounds and what I would call the tools and methods we need to a state of maturity that they're ready for trials, and those trials will then be more likely to succeed.
Perfect. And so how does that actually work in practice?
So the way we've looked at it and agreed across a very broad spectrum of the sort of dementia sector in academic, healthcare and industry sectors is three conditions really need to be met. The first is that the targets for treatments have to be shown to be relevant to the human conditions of dementia. So, and that's to get over one of the obstacles, that there are a lot of treatment compounds coming through very quickly that work in preclinical models, animal models, if you like, that then hit a brick wall when we try and bring them into people. So we need to make sure that the targets that our colleagues in the labs are working on also are valid for humans. That's the first condition. The second thing is that target in humans has not only exist in humans, but we want it to be directly connected to what we call the phenotype or the syndrome, the expression of disease, and the way that changes over time. Because if that target, the thing that those, let's say, a new drug might be acting on, if it is connected to the syndrome and progression, then treating that target or aiming at it is more likely to alter the course of the illness, either to slow it down or to prevent it. So that's the second condition. And the third thing is actually we need some new, rather specialist tools or ways, experimental facilities or scans and things like that. We need new tools to quantify what's changing and happening in the human brain, not at post-mortem, but actually early in disease when our participants and ourselves were very much alive and kicking, but with the early stages of these diseases, or even just at risk of them. Maybe it hasn't shown up yet, but we need some rather special new ways to measure that, and particularly ways to measure whether a new treatment is engaging in the target, whether it's hitting its target in the human brain in the way that we would like it to, and whether we can see early on that it's changing the course of the illness. If we can do that in a way that is safe and cost-effective and quick, we can accelerate the trials process. So you don't want to have to say a trial needs 1,000 people over 5 years to show it's going to work. It'll just never happen in reality. It's too big, too cumbersome, too expensive. What we'd like is to use these new specialist tools to be able to say in a short time, 3, 6, maybe 12 months, whether the new drug or the treatment is hitting the spot in the brain in a way that's doing something in the right direction. So we can make a decision. Is this a drug that should be pursued, or is it a drug we should call it a day on and focus our efforts and resources elsewhere? And I think that's that ability to get early signals, we call it, early signals of a drug being effective is so important. And I suppose that's why I call ourselves an incubator for these projects, not as a big clinical trials unit. Is to really show proof of concept, proof of probability that something is going to work in order to then focus investment. And I don't just mean the financial aspect of investment, I mean time, because every year that passes is very precious to families waiting for treatment. And also the effort of participants. It's a big thing to take part in a trial, and you want to know that the trials that you're being invited to have the best chance of success. We all know that success isn't guaranteed. We want to try and back the winners in the most effective way. So that's sort of the aims and sort of the way that we're thinking in the incubator across academic and industrial partners.
So it sounds like almost a filter between the ideas and the clinical trials to weed out any that are less concrete than others.
Yes, and as a filter, though, it's not just an ideas. It's not just talking shop and thinking through. It's with evidence. We want evidence to say this one has a good chance that it's doing the right thing. It's hitting the spot in the human brain. It's slowing the disease process down in people. It may or may not work at big scale into a big clinical trial, but it's something to back. And maybe another compound is something which we say it was a good idea. It was great to bring it forward, but let's call it quits. Yeah, and that's not a failure. It's a success if you know when to stop at the right time. I think it's really important people understand that knowing when to stop is a success, however disappointing for those who initiated perhaps that line of inquiry. Nobody wants to go down a blind alley and waste time, money, years, patient effort. It's something that's not likely to succeed. So it's a success to be able to say, this one, we're going to stop here.
I think that's really important to define what you mean by success, because, like you say, if you're wasting valuable funds, that will never be a success. So helping to eliminate the risk of that is so important.
Yeah, and it's a good word, risk. So we often talk about the role of the incubator is to de-risk clinical trials. And this concept of de-risking is really quite important, so that those that are looking to either invest in the trial or to participate, know that it's more likely to succeed. And I don't mean risk in terms of safety. I mean risk in terms of will it deliver the goods? Will it be shown to be successful?
I think that's a really good way to summarise it. What research areas does the EMI focus on?
The three active themes within the EMI that are ongoing, which I think cover some really hot topics in the dementia field at the moment, things that are really ripe for development with very exciting indicators of new treatments ahead. And these are looking at the role of inflammation in the brain in people with dementia or people who are at risk of later developing dementia. There's a sort of relatively low-grade background inflammation that appears to facilitate and accelerate the route towards dementia. So the neuroimmunology group is one area of tremendous interest. The other is of vascular health, and that takes the idea that not only do strokes affect cognition, but actually a much more pervasive vascular impairment is part of many dementias. Typically, you might see that it's small vessel disease, but even in Alzheimer's disease, it's not just about the amyloid plaques and the tau. There's a strong vascular element to Alzheimer's disease. There's many points of overlap between vascular health and Alzheimer's development. So the third area within the EMI is of synaptic health. And the attitude here is slightly perhaps less familiar term to many people, but the idea is that the synapse, the connection between two brain cells, is critical for doing what the brain has to do to get information exchanged, to have brain regions talking to each other, to store information and pass it on. This really depends on these connections called synapses. And these synapses disappear with dementias. They disappear quite early, in fact. They start to disappear even before brain shrinkage, we think. So it's a very early part of the disease process. And one of the reasons they disappear is that these junk proteins that your listeners will be familiar with, amyloid and tau, as they start to clump together, they're actually very toxic to the synapses. And then even worse than that, the inflammation that I mentioned is also directly damaging to synapses. So the synapse becomes a real battleground for the health of the brain. And it also is a way that one can try and get in to protect what matters for brain function, about the health of those networks and the connectivity in the brain for storing and transferring memories and thoughts and language and perception. And there's a number of ways that one might try to protect or preserve this flexibility, what we call plasticity, the adaptiveness of synapses, and their longevity. The other thing that synapses are important in is they're a very sensitive way to understand the health of the brain. A bit like listening to, the sound of a forest, the chatter in a forest, the bird song. It's not just any given bird, but it gives you an insight into the general health of the whole ecosystem. And so we've tools now to measure what synapses are doing and how their cells are talking to each other through the synapse as a way of indicating brain health. And that gives you a platform to try many, many different types of treatment in a very sensitive way. So synaptic health is, I think, a really important and emerging aspect of the world of dementia research, aiming towards better and more effective treatments. So those three things, vascular, synaptic, and the immunology or inflammation of the brain, are the three leading parts that are funded already, that are up and running in the EMI.
How could researchers get involved in the EMI?
We're very open to new ideas. So if there's researchers listening to this who feel that their work could connect with those themes, vascular, synaptic, and immunology, they should please come forward and contact us. Easy to find us through the Dementias Platform UK website, and connect to us, and we can put you in touch with the right teams and talk about what we're doing and how we can engage with new researchers. It's very much an open forum, so we're certainly not a closed shop. We welcome additional investigators. If you feel that those three themes of vascular, synaptic, and immunological health don't speak to your research, also come and talk to us. We're interested in developing other major threads, particularly, for example, sleep, and the importance of sleep to brain health, both as a sign of incipient dementia, perhaps even causing or accelerating cognitive impairment, but also as a route to better care and understanding treatment. So the whole way that sleep is part of good brain health is evolving very rapidly. And that may be something we'll see more of through the Dementias Platform UK EMI, in partnership with other people in the field.
Amazing. These three strands, do you have any kind of tangible examples of projects within those strands that have already taken place or are taking place?
Yes, so this one of the projects is called NTAD, or New Therapeutics in Alzheimer's Disease. I think there might be a separate podcast on this in a bit more detail, but it's a nice example. If I give a brief summary here, this is part of the work on synaptic health. We've got a number of ways to measure how healthy the synapses are, both by how many synapses there are, and this is through a special type of scan called a PET scan, which we can inject a dye that lights up wherever there are synapses, and we can measure for all parts of the brain how many synapses there are. But then we want to know how the synapses are working. And for that, we switch to a different method, something called magnetoencephalography, or MEG for short. Listeners may have seen the film The Meg. It's nothing like The Meg, the film. This is magnetoencephalography, MEG. It's a bit like EEG, which may be more familiar to listeners, but there are some technical reasons why we think it has clear advantages over EEG. And we're using MEG to measure how well synapses are being used to communicate from one region to another, not just in how as a thought or a memory is active, how that gives a sort of spark of activity through the brain networks, but also brain networks talk to each other really almost in a sing-song. They're called oscillations. It's a resonant frequency, more like an oscillation than a single burst. And these oscillations, it's a bit like the music of the brain, carries really important information around cognition and memory, and it helps the brain send the right information, not too much and not too little. It's a bit like both a filter and a channel. And we can measure these oscillations and the brain's activity with MEG with exquisite sensitivity. We typically record it 1,000 times a second. So we can see very fast changes, very fast exchanges through the brain. And it both gives sensitivity to the presence of disease, which we've shown through NTAD. We're currently looking at how sensitive it is to the progression of disease. And that's really as a foundation for using this methods for experimental medicines. And there's a number of examples, both connected with DPUK and out with DPUK, where MEG is used in conjunction with experimental treatments in a really exciting way. And what DPUK wants to do through the EMI and the project NTAD is to show that this MEG method, the MEG method, is reliable enough for clinical trials, that it could be scaled up. It could be at multiple centres and not just a super specialist skill at only one university, but actually can be undertaken across the UK. And above all else, that it's really valid for humans, that it does what it says on the tin. There's lots of work we're doing to show the validity, as well as the reliability and the scalability of MEG. So NTAD has been a really exciting project, and we're very far through the project. I haven't quite got the final results to talk, but I hope as people watch this space over the next 12 months, they'll be seeing and hearing more about the results from this NTAD study.
Brilliant, thank you. I'm sure that helps us all relate what you're saying to actual real life examples. So how does all of this ultimately help people with dementia?
The pathway to real impact, that line of sight to better prospects for people who either have dementia now or might have it in 10 years if nothing is done. There's always a bigger, more pressing problem, is that as we look to the future, 1 in 3 of us will get dementia if nothing is done. It's not just about academics and universities and pharma partners and industry. The third voice that's really important to us is the perspective of patients and their families, that the research we're doing focuses on the priorities that they also have. If you don't, it's not worth it. It's not what people need and want. So we have to make sure that we have this common goal and that we understand the priorities that patients and families have. In that, I'd include people who aren't yet patients, but are worried that they might become.
And I think that's reflected in the fact that you are so driven to make sure it's applicable to humans. All of this research that's come off great in preclinical models, like you say, the animal models, that they do actually have that practical benefit for the people who are the biggest stakeholders ultimately.
Yes, and we work very closely with our colleagues in laboratories, and we build on that work. But it doesn't stop. It mustn't stop at that point. It has to be brought into people, the patients, if you like that term, participants, human participants who are directly affected by the illness. We also want to, though, make sure that there's an open discussion between academic expertise, clinicians, and industry, so that there's that right sort of magic combination of knowledge and understanding of the disease, but also that any developments are going to be able to find their place in the real world of drug development and scaling up globally. And often that's through a commercial route. We're not linked to a single company, and none of these EMI projects are driven by a single company. This is what we call a pre-competitive forum. It's an open forum where we talk very directly with companies who talk directly to each other as well as to us. And that's very unusual in the research sector. And we've achieved that by developing communication and trust across our partners over recent years, and by recognizing that essentially we all face the same problem. If any one of us or any one of our companies were lucky enough to have a treatment, a magic bullet, you've then got the challenge to say, how can you show that it's effective? And that's a challenge we all face. And so for many of our partners who have compounds they're developing in the background, we're not locked into testing any one of those. We're facing a much more fundamental problem that is shared across multiple companies and multiple drug development processes.
Amazing. It's so important that universities and companies are working together collaboratively on this. Thank you so much for your time today. I think we will have a much better understanding about what the EMI is after that.
Thank you.
Thanks for listening to another DPUK podcast.